Hello बच्चों. Today we are going to start a new chapter, Principles of Inheritance.
As we begin Principles of Inheritance, let's ask ourselves, why is this important? Can anyone tell me why studying this matters?
Because this chapter helps us understand one of the most amazing questions of life, how traits are passed from parents to offspring, and why every individual is unique. Have you ever noticed that children often resemble their parents in eye color, height, hair type, or even smile? But at the same time, no two individuals are exactly alike. Why does this happen?
Let's start the chapter. What is genetics?
Genetics is the branch of biology that studies how characters are transferred from parents to offspring and why differences occur among individuals. This branch of biology that deals with the study of heredity and variation. Let's first understand the difference between heredity and inheritance, because many students get confused between these two terms. Heredity refers to the transmission of genetic characters or traits from parents to their offspring. It explains the resemblance between parents and children. Inheritance is the actual biological process through which traits are passed from one generation to the next through genes. Example, suppose a child has curly hair like their mother. The passing of the curly hair trait from mother to child is called inheritance. The similarity observed between the mother and child due to this trait is called heredity. So, in simple words, inheritance is the process, while heredity is the result of that process. Now let's talk about variation. Variation refers to the differences found among individuals of the same species. It makes every individual unique, even among members of the same family. For example, two siblings may have different heights or skin tones. One child may have straight hair, while another has curly hair. In humans, fingerprints of every person are different. Variation occurs because traits are inherited in different combinations from parents, and sometimes due to environmental factors as well. Variation is very important because it helps organisms adapt and survive in changing environments. Without variation, evolution would not be possible. Now, you may wonder, why are we discussing DNA and genes in this chapter? The reason is simple. DNA and genes are the basic units responsible for inheritance. They carry all the information that determines our characters and traits. DNA, deoxyribonucleic acid, is the hereditary material present inside our cells. It stores all the genetic information required for the growth, functioning, and reproduction of an organism. Gene is a specific segment of DNA that controls a particular trait or character. In simple words, genes are the functional units of heredity. For example, a gene may control eye color. Another gene may influence height or blood group. So, when traits are passed from parents to offspring, it is actually the genes carried on DNA that are being transmitted. Therefore, to understand inheritance and variation properly, we must first understand DNA and genes, the foundation of genetics. Before Gregor Mendel gave the scientific explanation of inheritance, people had many different ideas about how traits were passed from parents to offspring. This period is known as the Pre-Mendelian era. At that time, there was no knowledge of genes or DNA, so scientists and philosophers tried to explain inheritance based on observations and assumptions. Some believed that parental traits mixed like fluids, while others thought that a tiny, fully formed baby already existed inside reproductive cells. Although most of these theories were incorrect, they were very important because they laid the foundation for future studies in genetics. Later, Mendel's experiments provided scientific evidence and completely changed our understanding of inheritance. So, to understand why Mendel's work became revolutionary, we must first look at the ideas and theories that existed before him. Theories of blending inheritance, Pre-Mendelian theories. Moist vapor theory, Pythagoras. Fluid theory, Aristotle. Preformation theory, Jan Swammerdam. Particulate theory, Pierre-Louis Maupertuis. Theory of pangenesis, Charles Darwin. Theory of continuity of germplasm, August Weismann. Moist vapor theory, Pythagoras. According to this theory, every organ of the male body produced tiny particles, or moist vapors. These vapors collected in the semen and were transferred to the female during reproduction. The offspring developed characters according to the vapors received from different body parts of the father. This theory was incorrect because inheritance does not occur through vapors, but through genes present in DNA. Fluid theory, Aristotle. Aristotle believed that both male and female produced special reproductive fluids. These fluids mixed together during reproduction, and the blending of these fluids formed the characteristics of the offspring. For example, if one parent was tall and the other short, the child would show a mixed or intermediate character. This idea was later rejected because traits do not simply blend permanently. They are inherited through discrete units called genes. Preformation theory, Jan Swammerdam. This theory suggested that a tiny, fully formed miniature human called a homunculus was already present inside the sperm or egg. During development, this miniature individual simply grew larger in size without forming new structures. Some scientists believed the miniature human was present in the sperm, while others believed it was present in the egg. This theory was incorrect because embryonic development actually occurs gradually through cell division and differentiation. Particulate theory, Pierre-Louis Maupertuis. According to the particulate theory, hereditary characters are controlled by tiny, separate particles that are passed from parents to offspring. These particles retain their individuality and do not blend completely. Maupertuis suggested that both parents contribute equally to the inheritance of traits, and the combination of these particles determines the characteristics of the offspring. For example, a child may inherit eye color from one parent and hair texture from the other because different hereditary particles control different traits. This idea was much closer to modern genetics because it supported the concept that traits are inherited through distinct units, which we now call genes. Theory of pangenesis, Charles Darwin. According to the theory of pangenesis, all body cells of an organism produce tiny particles called gemmules. These gemmules carry hereditary information and travel to the reproductive organs, where they are passed on to the offspring through gametes. Darwin believed that the offspring develops by the fusion of gemmules received from both parents. Because gemmules were thought to come from all parts of the body, this theory also tried to explain the inheritance of acquired characters. For example, if a blacksmith developed strong muscles during life, Darwin's theory suggested that gemmules from those muscles could be transmitted to the next generation. This theory was later rejected because acquired characters are not inherited in this manner. Modern genetics proved that inheritance occurs through genes present on DNA. Now that we have understood the early theories of inheritance, it is time to discuss the scientist who gave the first scientific explanation of heredity, Gregor Mendel. Introduction of Gregor Mendel. Gregor Mendel was an Austrian monk, teacher, and scientist who is known as the father of genetics. He performed a series of experiments on garden pea plants, Pisum sativum, to understand how traits are passed from parents to offspring. Mendel carefully observed the inheritance of characters, such as plant height, flower color, seed shape, and seed color. By analyzing the results mathematically, he discovered that traits are inherited through specific hereditary factors, now known as genes. Why is Mendel called the father of genetics? Mendel is regarded as the father of genetics because he gave the first scientific explanation of inheritance. He proved that characters are controlled by discrete units, genes, and do not simply blend. He proposed fundamental laws of inheritance: law of dominance, law of segregation, law of independent assortment. His discoveries became the foundation of modern genetics. Even today, modern genetics is based on the principles discovered by Mendel. Students, Mendel could have selected many plants for his experiments, but why do you think he specifically chose the garden pea plant, Pisum sativum, as his experimental model? Mendel's experimental material, garden pea, Pisum sativum. Gregor Mendel selected the garden pea plant, Pisum sativum, for his experiments on inheritance. Why did Mendel choose the garden pea plant? Mendel selected pea plants because they had several advantages. Easy to grow and maintain. Pea plants can be grown easily in a small area and require less care. Short life cycle. They complete their life cycle quickly, allowing Mendel to observe many generations in a short time. Presence of clear contrasting characters. Pea plants showed easily observable contrasting traits, such as tall/dwarf, yellow/green seeds, round/wrinkled seeds. Bisexual flowers. Pea flowers contain both male and female reproductive organs, allowing self-pollination. Can undergo self-pollination as well as cross-pollination. This helped Mendel control breeding experiments easily. Pure breeding lines were available. Mendel could obtain plants showing the same trait generation after generation. Produces a large number of offspring. Large sample size helped Mendel obtain accurate results. These features made the garden pea an ideal organism for studying inheritance. Now, students, after selecting the garden pea plant as his experimental material, Gregor Mendel carefully chose 7 important characters for his study. Remember one thing very clearly. Character means a heritable feature being studied. Trait means the different forms of that character. For example, plant height is a character. Tall and dwarf are its traits. Now, let's understand Mendel's 7 characters one by one. 7.1. Plant height. Traits, tall and dwarf. Some pea plants were very tall, while some were short or dwarf. 7.2. Flower color. Traits, violet or purple, and white. Mendel observed that some flowers were purple, whereas others were white. 7.3. Flower position. Traits, axial and terminal. Axial flowers grow along the sides of the stem. Terminal flowers grow at the tip of the stem. 7.4. Seed shape. Traits, round and wrinkled. Some seeds were smooth and round, while others appeared wrinkled. 7.5. Seed color. Traits, yellow and green. Seed showed either yellow color or green color. 7.6. Pod shape. Traits, inflated and constricted. Some pods looked full and swollen, while others were narrow and constricted. 7.7. Pod color. Traits, green and yellow. Pods were either green in color or yellow. Mendel students. These characters were very easy to identify and showed clear contrasting forms. This helped Mendel observe inheritance patterns accurately and formulate the laws of genetics. Now, students, let's understand the techniques used by Gregor Mendel in his pea plant experiments. Mendel followed a very systematic and controlled method to study inheritance. Step 1. Selection of pure parent plants. Mendel first selected true breeding, pure pea plants, having contrasting traits. For example, pure tall plant times pure dwarf plant, pure yellow seeds times pure green seeds. These plants produce the same traits generation after generation. Step 2. Emasculation. In bisexual flowers, both male and female reproductive organs are present. To prevent self-pollination, Mendel carefully removed the stamens, male reproductive parts, from the flower of the female parent before the anthers released pollen. This process is called emasculation. Step 3. Artificial pollination, cross-pollination. After emasculation, Mendel transferred pollen grains from the selected male parent to the stigma of the female flower manually. This controlled transfer of pollen is called artificial pollination, or cross-pollination. Step 4. Bagging. After pollination, the flower was covered with a bag to prevent unwanted pollen grains from entering. This step ensured that only the desired cross took place. This process is known as bagging. Step 5. Formation of pods and seeds. After successful pollination and fertilization, pods and seeds developed. These seeds formed the F1 generation, first filial generation. Step 6. Self-pollination of F1 generation. Mendel then allowed the F1 plants to self-pollinate and collected the seeds produced. These seeds gave rise to the F2 generation, second filial generation. Step 7. Observation and analysis. Mendel carefully counted and recorded the number of plants showing different traits in each generation. His accurate observations and mathematical analysis helped him discover the fundamental laws of inheritance. This systematic approach made Mendel's experiments reliable and revolutionary in the field of genetics. Thank you, students, for attending today's lecture. Today, we learned about genetics, heredity and variation, pre-Mendelian theories of inheritance, introduction to Gregor Mendel, why Mendel selected pea plants for his experiments, Mendel's 7 contrasting characters and experimental techniques. In the next class, we will discuss Mendel's experiments in detail and understand how he discovered the fundamental laws of inheritance. Until then, revise today's concepts carefully and make sure you understand the basic terminology, as it will help you grasp the upcoming topics more effectively. If you found this lecture helpful, don't forget to like, share, and subscribe to the channel. Also, leave your questions and feedback in the comment section. Your comments help us create better content for you. Turn on the notification bell so you never miss an important biology lecture. Thank you once again, and I'll see you in the next class. Happy learning and keep exploring biology. Bye-bye and take care.